Okay, remember that chapter 31 is not going to be on an exam. I mean, if you look at exam number 4, the very last one we take, it's chapters 33 to 34. Oh, wait. That should say 32 to 34. What the heck? Yes. Yes, your last exam, I got to make that correction. That should be 32 to 34. Your last exam is over all the animals, which is in those 3 chapters, 32 to 34. Right? Fungi. Plants, 32. Animals. Right, okay. Anyway, back to chapter 31. I'll make the corrected syllabus and I'll post an announcement about that thing with the last exam if you didn't get exactly what I was saying, but I'll put that on there. Anyway, chapter 31 on the fungus, I do not put on an exam. It won't be on the last exam. It's not on the exam on Tuesday. The exam on Tuesday is just plants. It's 2 chapters, but it's just plants, seedless and seeded plants. But you do have an additional assignment, a chapter 31 assignment. Maybe I call it a quiz or something like that on Blackboard. Make sure you look at the spreadsheet for chapter 31. Actually, he's actually being good today, so he's sleeping, that's why. Make sure you look at the spreadsheet and understand what's due for chapter 31. There's I think that's it. I don't have it in front of me, so I don't want to say anything. But if you have any confusion, I think there's a dynamic well, I can just look at the grade book. I can tell you what's due for 31. Hold on. Let's settle this right now so nobody's confused. Okay, so you have a homework from 31. You have a dynamic study module from 31. And you have a quiz. That's the new thing. You have a quiz. Yeah, 80 points. You're right, Jennifer. Don't forget to do that. I don't think it's timed. I rarely time my quizzes because inevitably what happens is one of you is sitting at their computer and the internet drops out. And then it's this thing of like, you could use that to say that happened. Not that I think any of you guys would do that, but it's still one of those things where I don't make it timed. But, and of course it's open book. I'm not going to expect you to say, well, Santa might know. All right, any questions on that about chapter 31 and the assignment? So not on the exam coming up on Tuesday, not on the exam at the end of on the semester. There will be questions on the lab practical about fungi because we are going to do a lab on fungi. We're going to do a lecture too because I wouldn't resist talking about these things. Okay, let's talk about the fungus among us. Isn't that funny? My dad would always say that. The fungus among us. I'm like, yeah, it's really, really funny, Dad. Especially when you know how your parents are, right? They say the same funny thing a million times. These are the last few folks that came in. Sydney, Spencer, hi. If I didn't say hi, I'm sorry. Let me see who else is here. Marcelo, Jennifer. I think I got everybody. Okay, good deal. Remember, there's a field trip tomorrow. Looks like the weather's going to be decent, too, so that's kind of cool. So if you want to go, I had one more person, so we're up to, 10 now, I think, or 11. So we're getting to the point where, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, yeah, something like that. We're getting to the point, though, remember, we can only take so many cars. I'm bringing my wife's Honda. She has a CRV. I'll have to sneak it off, though, because she'll get mad if I take it. She always, many times sometimes when I come home, I have a dead animal in my car or my truck with me. I'm not going to pick up a raccoon. It's not because I'm hungry or anything. But like if I see a dead bobcat, I'll pick it up because I know somebody does work on bobcats. I haven't heard because the other professor with his class, he was supposed to, that reminded me. Let me email him. That was a professor in another class. He told me he was going to do that, but let me write a note. But Thomas, you're driving yourself anyway, right? And what do you drive? How many folks can you fit in your car comfortably? Now, well, I got to think about they have to come back. So, and you're not coming back. So, anyway. But my wife's CRV, I'll bring that. But like I said, she doesn't like it when I come home with dead animals in it. Well, like I said, if it's something, if I was going down the road and I saw a dead, well, if it was a dead panther, probably not, because I couldn't get it in the back of the car. But a dead bobcat, I would, because like I said, I could come home, put it in the freezer, and then later on we can put it in a crock pot. No, later on I can actually call my buddy up who'll come get him, and he does work on them. Research work. Okay, anybody ready to talk about fungus? You know what I like about fungus? What I like about fungus? What I like about fungus really is the fact that they can be so weird, so bizarre, because they really sort of don't follow the ant and animal and plant. Boy, I did one of them spoonerisms where you switch the syllables. Ant and planimal conventions. I just did it again, but anyway, of color. Okay, plants typically have to be green because they got to photosynthesize. Now they can play around with the flowers quite a bit, but you know the important part, they're going to have a good part of their body's going to have to be green, unless they're the red algae, right? Remember, they can do it, but even though they still do have chlorophyll. And animals, they can be of colors, right? But I'm trying to think, is there any really, other than with striping or thing? I'm trying to think, is there any really brilliantly colored? Yeah, some of the insects are. You know, I'm trying to think like any mammals. There's all kinds of stripes, but is there really just like is there like a lightning blue stripe somewhere? Probably one of the primates. I'm thinking one of the primates in the back of my head, probably like, what were they called? Oh, what are those one monkeys called? Yeah, right. Don't they have really, don't they have really pronounced, you could look it up online, like coloration, you know, around their faces, but for the most part, when you're dealing with animals, right, it's kind of the typical spectrum of colors. But what I like about fungi, back to that again, is the fact that they can come up with all these bizarre shapes and forms and colors. They're not limited by being aerodynamic. They're not limited by being hydrodynamic. They're not limited by having to grow tall for the sun. They just really kind of do their own thing. And another thing that's really, really fascinating to me is if you talk to people and you say, all right, even if they don't know the kingdoms of life, which I know you guys do, but if you talk to people and you say, all right, a fungus, a mushroom, a dog, and let's say an oak tree, okay, a mushroom, a dog, or it could be you, whatever, an animal, a mushroom, a dog, and an oak tree. Who of those three, going back to that cladistics chapter, who of those three, which two of those are more closely related? Okay, so, or another way to say that, as an animal, am I closer related to a mushroom or to an oak tree? What do you guys think? Why? Can you give me a piece of evidence, or is that, I mean, it's a 50-50 guess, but, and you might know, but do you remember a piece of evidence that suggests that? Or does anybody have evidence that it's really a plant? I mean, who's our closest relative? You know, go back to some characteristics that we share. Is there something that might unite us all together? More closely than, it's just the Sesame Street routine. One of these is not like the other. Is it the animal, or is it the plant, or the mushroom? Okay, well, I'm going to leave you on the edge of your seats then. I'm not going to tell you till we get towards the end and start talking about the taxonomy and how fungi come into the place here, okay? Another thing I like about mushrooms, like I said, is the fact of the way the coloration is arranged. Look at these mushrooms on the right. These are turkey tail. They grow in Florida. You probably have seen them. There's all kinds of, and that's another thing. Mushrooms are a real pain as far as identifying, because again, they don't really have to abide by a whole lot when it comes to shape and color and all that. But turkey tail looks that way, I guess, because if you looked at the tail of a turkey, when it's opened up, the color of the feathers, you sort of get that view. Although don't be confused, there are lots of things that are known as bracket fungus. This is a bracket fungus. It grows like on a tree, like a shelf. That's the bracket, but not all bracket fungus are turkey tails. Okay, another really, to me, fascinating thing about mushrooms, and this one I will give you the answer to, and this is like something I remember sometimes going to middle schools and talking to kids, and it's always like, what's the largest organism what's the largest animal in the world? And I'm like, well, it's probably the blue whale. Okay. And then the other questions are usually things like, who would win between a killer whale and a shark? Okay, all right. But then I ask them, okay, guys, think about not just animals. What's the largest organism in the world? Now, they probably don't know much about bacteria or anything like that, but I mean, and many of them may not know that a fungus is a living organism. But if you look at the screen here, if you ask that question, you rarely, even with people in the know, who know that a fungus is a living thing, are going to know the right answer. They're going to say, oh, it must be the blue whale or those redwood trees. No, it's actually a mushroom, and it's actually a mushroom that has a species that grows here in Florida. They're called the honey mushrooms. Oftentimes, if students come up to me and they show me a mushroom, if it's any sort of honey-colored mushroom, I go honey mushroom. Start with that, because there's lots of different kinds of honey mushrooms. Honey mushrooms, Armillaria, that's the genus that's up there, Oystiae. So there's lots of different kinds of species of these. But this organism, not the ones in Florida, but there's one out in Oregon. One, right? There's an organism, there's a honey mushroom individual out in Oregon that covers somewhere in the vicinity of 9 million square miles of forests. It's like, oh, I was looking it up online. I sent you the link at the very beginning. Yeah, that's it. Click on that first link at the very top of the chat. If you see it, I think I still have it pulled up on mine too. Yeah, it is. It's 2.4 miles across in the Blue Mountains in Oregon. 2.4 miles across. Now, if you're on that website and you scroll down, this is a honey mushroom species, or it's the same genus, right? But you'll notice that what you're looking at is the typical toadstool. Now, we're not saying, and that was the good part about Spencer's question there. We're not saying that you're walking through the forest there in Oregon, you come across this toadstool that's 2.5 miles across. No, that's not what we're saying. You see, the thing about mushrooms is most of a mushroom is underground or hidden. These things that you see that we call toadstools, those are just the fruiting bodies, which is kind of another way of saying the reproductive parts. So when these things appear, they appear from subterranean, underground. They pop up. They can pop up pretty quick. You know they do, right? You wake up in the morning like, wow, the whole yard seems to be covered in mushrooms. Must have been all these spores that blew in. Even if you know mushrooms have spores, it's probably not that. It's a reason that we'll talk about later on when we go through how these organisms grow. There's about 1.5 million fungal species, so they're certainly not the biggest group. I got a cool little pie chart. Look at the biggest group, the insects. Right? If you wonder where we're fitting in, oh, don't grab that pen. If you wonder where we're fitting in, we're over here with the frog on the left. That's our slice of the pie. Pen dead? The other one. There we go. We're right over here. So I said with that little tiny sliver. Yeah, that is the littlest sliver, isn't it? Number of species. So it would be diversity. Yeah, but that's a good question to ask too, Spencer, because when you look at charts like that, yeah, there's two very different interpretations that can occur. In this particular case, we're looking at how many species contributed, so it's diversity and not total, like you said, total biomass. Good question. Okay, so the fungi, though, I mean, they got a fairly good slice of the pie. If you think about it, it looks to me like they're almost about second place, right? Arachnids. Oh, man, I love arachnids. Anyway, they're definitely third. Plenty of bacteria. But so there's definitely a lot of diversity among these guys. And that's because, as I said, they really don't have a lot of confines. They really don't obey a lot of they obey all the rules of life, but they're really sort of that's the bacteria. They're sort of just pushing the limits of what you can look like if you're living, I guess is the best way to say it. Some of them are single-celled, some are multi-celled, some are both. It's not really about the cell in fungi, okay? You know, we always talk about the cell theory, the cell doctrine. cell division, cell reproduction, cellular structure, cell. In fungus, it's really more about the nucleus. It's really more about the genetic material, okay? So I'll talk a little more about that later on, but that helps if you, when we go through this, how you can understand the structure of fungus. If you think about it, it's really not, they're really not focusing on the cell like we do in all these other creatures that we talk about. They reproduce sexually, although typically they're going to be asexual reproducers, okay? Okay, now, one of the things that is really different about them is the fact that they undergo digestion. So that's not the different part. Every organism has to break down bigger molecules into smaller ones to release energy to do the respiration thing, right? All that thing you learned in Bio 1. Now, you know, we do that, us animals, for example, we do that by bringing food into our body, right? We eat, for example, in humans, we eat, we bring food into our stomach, we tear it apart chemically, blah, blah, blah. But we eventually are trying to get at those little glucose nuggets because we're trying to pump those into the mitochondria, right? So we do what's called intracellular digestion. Now, what that means is inside of our cells is where that actual glucose being used to make ATP is happening. Outside of our cells, in our stomach, is where we're topping down the stuff, but that actual energy extraction doesn't occur until you're in the mitochondria or in the cytoplasm if you're doing glycolysis, right? So again, this is Bio 1 stuff. Now, what fungi do that are different is they do something called extracellular digestion. So they have enzymes like you and I, and they really are technically enzymes, but just to kind of emphasize the difference, we call them exozymes. See, what these guys do, what these fungi do is they secrete enzymes into the environment. They secrete these digestive enzymes that are in our stomach, that are in our intestines. They make those, and then they pump into the surrounding environment. They digest all the organic material. That's why it's always good, you know, living underground. It's the best place to live. So they digest all this organic material, like our stomach would do, like our intestines would do, and then they just kind of, they kind of swim in it. Yay! It's like that one commercial for the insurance company that love the Meermuts, where the dogs turn on the water faucet, flood the house, and then have a synchronized swimming thing. You have no idea what I'm talking about, then I'm sorry, but it's a funny commercial. Okay, so that's what we got going on with the mushroom. Now, that isn't the fruiting part that you see in the background there. That's another part that we'll talk about, but that's a big thing. That's a big thing because if you do remember, I have mentioned to you more than once about plants, that plants moved out onto land, the first ones to move out on the land, but they did so in conjunction with the fungus that we'll look at it again in this chapter, right? And this ability of the fungus to extracellularly digest, to make this soup in the soil, this soup of or digesting organic materials, some materials that only fungi can digest. Some materials on Earth that are organic, that are so strong that you need a fungus to tear them down. When the fungus are able to do that, the plants more easily, more readily are able to suck those nutrients up and use those to grow. Because, of course, the fungus is secreting those exozymes for itself, but you're digesting more than you're absorbing. So there's an excess. And the plants go, wow, this is perfect. This is nutrient soil. It was before, but not quite in the form that plants could use. That's what the fungi did for it. Okay, it's us and the animals. Us and the animals. Us and the animals. I am an animal. It's us and the fungi is what I'm trying to say. We're the ones that are more closely related than a plant. We shared an ancestor. Look at right around when that Cambrian explosion thing was going on, right? Remember, it wasn't an explosion like one day. It was an explosion over several million years. So, you know, and remember what I said was going on back then? That's when Darwin said, whoa, where did all this stuff come from? So it's about that time we get that split. We get a lot of splits going on back then. One of them is us and our fungal ancestors. We're more closely related to a fungus, believe it or not. Food. In function. Yeah, in function. The way they can evert their stomach into the mantle of a mollusk, yeah, in function, sure. I guess the one major difference would be that they're going to digest all of the mollusk, right, and not actually just you know, make a soup of it. So, yeah. Okay, now how do these things? Here's another bracket fungus. Some of them are really pretty. That's why I said it'd be beautiful orange colors and yellow. I mean, they're really fascinating because of this whole living outside the box when it comes to color. So these are some fungi growing on a tree, and as I said, of course, they have exozymes. Now, I'll show you in just a minute what a mycelium is, but the mycelium, if you want to kind of get a visual right now, that's what's anchoring this bracket fungus to the tree. And that's what would be the, and this is not the right word, but in function, okay, the roots that hold the toadstools into the ground. If you were to very carefully pull up a toadstool, a mushroom, you might see these little white fibrous things. You might think they were roots. And they're not, though, right? They're not quite the same, because roots are things that are found in plants. It's actually a part of that mycelium. And again, I'll talk more about that. But the mycelium work very much like the roots, okay? Because what they do is they hold this mushroom onto this tree or onto the ground, and they break down organic material. It's these little fibrous things that are secreting those exozymes. Okay, so that's where it's a little different than plants, right? Plants aren't doing that. Plants are sucking up those nutrients. Plants are digesting food intracellularly like you and I do. They have mitochondria, okay? These organisms are doing a little bit differently than that. They're using these exozymes to start the digestion process. They can break down cellulose. They can break down that stuff you and I can't break down. They can break down that stuff that Bessie the cow, yeah, needs those symbiotic bacteria in her gut to break down. That's a durable organic material, a lot of carbon. Don't want that lost. You want these decomposers. See, you're getting it right. Fungus are decomposers. This is lignin. It's what makes wood hard, which is a very simple way to put it. But it's a very complex organic molecule. Fungi are one of the only known organisms that can break that down. See, animals can't digest either one of those. So man, if all that carbon and all that material is lost to the environment, remember, we're all connected. We're all connected because the carbon atoms in your body, well, they're actually made in an exploding star, and some of the carbon atoms in your body might be carbon atoms that were in Abraham Lincoln's body, because he decomposed and carbon goes back in the environment, and the recycling thing that we talked about. And some are carnivorous. There are carnivorous fungi. Yes, there are. Yes, there are. It's so cool. I'll get to those. I'll show you those. All right, let me go back to mycelium for a minute so you understand what I'm talking about. Okay, mycelium, little root-like things. Here's what the mycelium look like up close. Mycelium is a bunch or a mat, M-A-T, of these things called hyphae. Those little root-like things, those little individual root-like things, like you might even think if you pulled up a toadstool, if you kind of remember, it didn't really look roots, but more like a net, maybe, a little net. And that's because a net is a collection of strings. We call those parasitic, Spencer. Yeah, and I mean, in terms of the effect, it's the same. If you're a carnivore, the effect on the prey is the same, but we probably call those more parasitic. It's kind of like that that brain-eating amoeba. Call that carnivorous, right? Yeah. Okay, so these little hyphae that make up that mat that we're talking about, and I'll show you some pictures if you're not getting a mental, but these mats are made up of these long little slender things. So here's kind of the root-like things called hyphae, and they're filaments. They're either, and notice what it says right here. That's what I'm talking about. It's not about cells in these guys. These filaments are either cell or cells. All right, they found in multicellular fungi. Well, wait a minute. How can you be just a cell but be multicellular? Well, look down below. There's two types of hyphae. On the left-hand side, there's the kind we call septa or septate. And the reason why it's called septate is because a septum is a division. But, notice that over here on the far right, see in that picture? Even though there is a division, there's an opening, there's a pore, so things can go through. but maybe more slowly, maybe more controlled, right? That's kind of the thing there. And the little purple things that you see in septate hyphae, sometimes, not always, that's going to be just a single nucleus. Sometimes it's going to be a double nucleus, as we'll see when I get to some of this weird stuff that they do. Now, the other type is the weird cell multicellular. Fungi are multicellular, okay? They're considered multicellular. Not the same multicellular as in animals. I'm sorry to put that in there. I just want to remind you, but they are. But hyphae that are synocytic, that's how you say that word, synocytic. Synocytic hyphae are one cell, several nuclei. So that's kind of where the multicellularity thing comes up, especially in fungi. Remember, not about the cells, it's about the nuclei. I got bunches of nuclei, so I'm multinucleate is another word we can use to refer to it, but they're basically kind of multicellular in function, because that's really what a cell is a bunch of different nuclei with supporting structures, kind of, if you think about it that way. Now, what's the advantage of having these hyphae? Whether you're septate or synocytic, the advantage is you can do something now. You can grow now. All right, you're living underground, you're going through exozymes, your mycelium are spreading out, and all of a sudden conditions become really ripe for having babies. Honey, let's have some babies, right? The fungi say to each other. Now, right, so they're all underground, so it rains one night. That's where those mushrooms come. When you come up in the morning, you go, where all these mushrooms come from? Boom, all those hyphae swell, like cytoplasm just gets pumped to the end of the hyphae. They swell, and they become a toadstool. They pop up. And on those toadstools, we're going to find out there's gills, and there's spores on those gills, and the spores go drifting off into the environment. And you're going to do that because you're going to say, wow, it's great time to have kids, because these spores have a better chance of germinating when it's moist out. Fungi tend to be like ferns that way, or mosses. They like moist environments, although there are some exceptions. All right? Now, sometimes organisms engage in reproduction. You know, I know if you're thinking, wait a minute, I thought organisms engage in reproduction when it's harsh out. They do. Sometimes they engage in reproduction when it's beneficial to send out spores. Sometimes they engage in reproduction when they feel it's beneficial to generate genetic diversity. Okay, so you see, and species of organisms can kind of, engage in both behaviors, or one species does it one way, one does it another. All right, reproduction is either yay, everything's great, or whoa, we better start saving our genetic material for the future. Better start generating some variation. We don't know what's going to happen. Remember, Mother Nature changes and throws the cards out, and the cards are even different decks of cards each time. One time you're playing with a regular deck of cards, and then you're playing Uno, that card game. So got to be ready to do stuff now. Boom, flow that cytoplasm, popped up those toadstools, crank out those spores. Things are good. It may not rain tomorrow, or maybe tomorrow is going to be worse or better for the environment. All right. Here's what I mean by the mycelium. See the hyphae? The hyphae are the little threads there, and a bunch of them is a mycelium. So if you're wondering how, maybe not, but if you're wondering how the whole mycelial mat and exozyme thing works together, and if you're thinking surface area to volume ratio, you're right on the money. We could do the math here, but you remember in Bio 1, as cells stay smaller, small cells have a higher surface area to volume ratio. Now, does anybody remember why that's an advantage? Because cells are small. Why is it an advantage to have a high surface area to volume ratio? You remember, it increases the efficiency of something. You remember what that was? Go back and change your Bio 1 grades. But in this class, it was in my Bio 1 class. Let's see here. No, Carson was, but he's not here today. Come on, guys. Why do cells have a high surface area to volume ratio? Yeah, absorption. And the opposite of absorption would be secretion, right? So to increase the efficiency of things moving across the membrane. Now, you got a lot of surface area to volume ratio with little look on the right. Look at the little mycelium. Under the microscope, the scale is 60 microns. Look how small they get. But look how many there are. Really high surface area to volume ratio. Lots of absorption. Yeah, I got you. Lots of absorption, lots of diffusion, lots of osmosis. Very easy, very thin, little compartments with cells or a cell by which things can move across. Okay? Now, it just so happens having a lot of these hyphae built on the mycelium does help you stand up or anchor onto a tree too. Okay, so not quite like roots, right? Not the same as in roots because we're not doing the same kind of digestion here. Now, another interesting thing, everything, if you're going to study fungal anatomy, people who study fungus, by the way, are called mycologists. So if you ever see that word, like if your doctor said, I'm going to give you a drug that's going to treat a mycological infection, it's fungal. Yeah. Yeah, yeah, kind of. Other than the extracellular, intracellular part. But other than that, you're right. Same idea. Okay, so hyphae make up all of the mushroom. All the fungi we're going to talk about, they're made of hyphae. Everything is made of hyphae. There's stuff underground, which is the mycelial mat, which is doing the absorbing and a little bit of the anchoring. And the toadstool that pops up, all of it's made of the same thing. Next time you're having like raw mushrooms on a salad, tear them apart, and they tear apart in what's the word? Like they're fibrous kind of, right? They kind of tear apart in sections, vertical, like longer sections. You can sort of rip a mushroom. Does that make sense? We do the lab on the mushrooms. I'll bring some in from the store. We can see. I'll try to find some out in the woods. Sometimes I find some out in the woods too. Look at this picture here, 10 microns. See how small the hyphae get? And then in the back, look, I'm just showing the extensive mat they could make. The little picture in the middle, I think that's kind of cool. It looks like something splattered up on a wall. Then a little bigger microscope. But look at all that. That's all for absorption. It's all for diffusion. It's all for whatever's necessary. It's really going to help if you're an exozymer, isn't it, huh? Now, very interesting part. We talked about relationships, plant, animals, and fungus. And I asked you guys if you knew what was one of the criteria, what was one of the evidences of our closer relationship with a fungus. Now, remember that term close relative, distant relative, it's all very relative when we're talking about relationship. It's all very relative what you mean by distant. But one of the things that unites us is the walls, the cell walls made of, look, I got some cool pictures. These are cenocytic hyphae on the left. See, they're open. And then you can even see the septum in the ones that are purple. But the cell walls of all these things are made of chitin. You remember chitin, right? You remember chitin, don't you? Yeah. It's a carbohydrate, isn't it? It's one of those modified carbohydrates. There's a nitrogen group that's put on it. It's kind of the animal equivalent of cellulose. It builds the exoskeletons of insects and crustaceans, just like cellulose builds the cell walls of plants. Does anybody know what this is? A beetle found in Florida. You might find one for your field notebook. Does anybody know what that is? Arthropods are insects and crustaceans and things we'll talk about later. Does anybody know what that beetle is? It's not Ringo or John or Paul. Sure is. Male or female? How do you know? Thank you. And so that says something about male rhino beetles' behavior, doesn't it? What do you think that says about the behavior of rhino beetles? and maybe more specifically their reproductive behavior. What kind of behavior might you think they engage in? Typing? Oh, no. Okay. But heck, that'd be interesting if they could type. There. Yeah, there you go. Yeah, male combat. It's like being a detective, remember? So, again, next time you hear about some fossil flying and you go, how could they tell that? Think, well, maybe you don't know what the links are, but some expert might say, I know that because of this. You know? I had a buddy of mine who did work on dolphins where they fed. You know, where dolphins, we find out that dolphins have pretty good what we call site fidelity. They kind of stay in their little neighborhood. They might travel some, but they spend a lot of time in their own little neighborhood typically. And one of the ways that my buddy was able to determine that or support that claim was he was able to look at the fish they were eating, and the fish they were eating had different concentrates of this radioisotope, radioactive, but not damagingly just slightly radioactive. And so using a little detective work, he looked at the stomach contents of these dolphins that would wash up dead, and he was able to tell where they came from by what they had been eating by looking, oh, the fish that this dolphin are eating have this concentration of this radioisotope, and the fish that the other dolphin are eating have a different concentration. And when we go out in the wild and we collect fish, we say, oh, these fish that the dolphin we're feeding are over here, and these fish the dolphin we're feeding on are over there. Correct, intrasexular, right? Between male to male. Between male to male. Oh, wait, hold on. Oh, no, intra is outside. Intersexual. Intersexual is male to male. Intrasexular is females pick males. Hold on. Sorry. I always get that confused. Inter is outside the group. Intra is within the group. It doesn't help that when I first moved to Florida, holy cow, I was confused, especially Orlando. Because, you know, I knew what intra and inter meant at that time. And what we got run right through the middle of Orlando is Interstate 4. Is Interstate 4 even interstate? Is it really an interstate? Yeah, it's happened to Daytona, doesn't it? So I'm like, wait a minute, that's not interstate. All right, enough of that. Change the name. We should raise a petition. Here's the carnivorous ones. Little Mr. Nematode, microscopic worms, not worms like segmented worms, but worms only because of their shape. Years ago, they used to throw all the worms in the same group. There's that taxonomy stuff because of shape. And we found out that that was very paraphyletic, okay? Here's little nematode, microscopic, 25 micron is the meter that you see up there up above. Just making his way, minding his own way, moving through the environment, and here comes some hyphae. Now, hyphae means you got a fungus in the area. You know, if a doctor's examining you for some kind of infection, he finds hyphae or mycelium, he's going to say, oh, this is a fungal infection, okay? Oh, he may not say it that way. Oh, this is a fungal infection. But he's definitely going to say it's a fungal infection. And what the hyphae do is they can make a loop around Mr. Nematode, and very quickly, remember that if you got those open cells, boom, you're going to pump that cytoplasm into the end. You're going to wrap that loop in less than a second. You got yourself a nematode. You're going to wrap them up a couple times too, and then you're going to withdraw those hyphae, and then you're going to continue to wrap them up with more and more hyphae, kind of like a spider wraps up its prey in spider web. And of course, those hyphae are good for holding you to the ground. They're obviously good for catching worms. They're also good for absorbing. So you can start absorbing that worm right away. Probably not a great way to die if you're a nematode. Luckily they have no brain. And then you take the surface area to volume ratio stuff and you go to the next level when you infect plants, because the fungal hyphae, when they grow into plants, form these things called haustorium. Haustorium is a word that means something that does this. Okay? Forget it. I won't confuse you. You see what I mean? So that again, so you got surface area to volume ratio on top of surface area to volume ratio of the hyphae. Lots of absorption. And those can actually, penetrate into the wall of the cell. Sometimes even into the plasma cell membrane too. And it kind of depends what kind of fungus they are. And we'll talk not in all cases do you have a fungus parasitizing. It looks here, this is parasitism. Looks like it's parasitizing the plant. If it's puncturing, if it's puncturing into the cell plasma membrane, like the stuff on the left there, all of it actually, then it might be more inclined to be parasitic, because there's other type of fungi that live amongst the cells. Let me show you. These are the mycorrhizae that I've hinted to. This is a mutualistic relationship. Both get a benefit. Mycorrhizae, look on the right, is the little fuzziness on the root of that plant. That fuzziness is not little tiny roots. Sorry, what was the snake? Spencer, what was the snake you were telling me? I missed that. Chet, I'm sorry. Yeah, it's like there's a lot of snakes that are constrictors. There's one huge family of snakes that's called the constrictor family, and that's one of the ways they kill. Yeah, I always think that would be the worst way to go, being suffocated by something like that. But this is, I guess then this is the good fungus, because like I said, that's what that is right there. And that's going to increase surface area to volume ratio, very small, absorption, digestion. And what the plant does for the fungus is it helps provide it with a source of carbon. You need carbon to build molecules. So it kind of like is, hey, you help me digest this food. The fungus does it for itself, obviously. The root, the plant absorbs the rest, uses it to build up its carbon, has some excess carbon that says, hey, it's like a kickback. There you go, fungus, there's a little bit of a kickback. Now, if you're thinking, do they actually get down to the table and have a negotiation and shake hands? Of course they don't do that. But what they do is evolution through time has favored them to doing that to each other. You scratch my back, I'll scratch your back. You scratch my back, I'll scratch your back. And maybe we become, in the case of lichen that we'll talk about, it's become so bad they can't even live without each other. This is such an important mutualistic relationship that it's found, folks, on 90% of all vascular plant species roots. All vascular plants, so everything past the fern, right? Everything past the fern has 90% of that. They even did an experiment where they sprayed a fungicide on this plant, and they didn't spray one on this one. A fungicide would kill a fungus, bad and good. That's the problem with things like that. They kill the good and the bad. And you can see that this plant on the left, although it didn't die, it didn't grow as well as the plant on the right. Right? The difference is the one on the left had no mycorrhizae. You killed it with the fungicide. This one did. Just as a side to Indian pipe, this is a plant that we have growing in Florida, actually on campus in a couple of spots. I have to take a look and see if I see it again. Sometimes I see it in late October, November. But this is a flowering plant, but the plant part is probably the most interesting because it obviously doesn't photosynthesize. It's white. Well, what happens is the Indian pipe, which is the flowering plant, the roots of the Indian pipe actually live on the roots of like oak trees, and their roots, so they hold on to that, and their roots extend out into the mycelial mat, and they absorb some of the nutrients as well. So we say that it's a tripartite relationship because it involves basically kind of the roots of the Indian pipe parasitizing the fungal roots, okay, or entering into a relationship with the fungal roots, maybe not necessarily parasitizing, but that are in a relationship with the roots of the plant. So I'm trying to explain that a tripartite relationship involves three connections, not all of them plus and plus. The roots of the Indian pipe growing out into the mycelial mat, I've heard that called parasitic, but I'm not sure exactly what the harm does. other than removing some of the food. Maybe that's what it is. But yeah, isn't that cool? Don't even have to photosynthesize. Living things always find a way, man, I tell you. Okay, now back to mycorrhizae for a second. If you're going to be why am I having such a hard time with that word? If you're going to punch a hole into the plasma cell membrane, you're probably a parasitic fungus, okay? But you can also do it a different way. You can be, again, mycorrhizae, helpful fungus. And there are two types. Ectomycorrhizae do exactly what it says. And you can see in the picture, this is a cross-section of a root, okay? And the green stuff on the outside is the mycorrhizae. It grows ecto on the outside. The hyphae of ectomycorrhizae, down in the red, I wrote it for you. It surrounds the roots and it goes between the cells. So it goes between the cell membranes. Here's the microscope picture you'll see in class when we do the lab. All the part here is mycorrhizae. Look at that. It like coats it, doesn't it? This is all plant. You should recognize the vascular tissue, the xylem and the phloem. It looks like a hair, a hairy root is what it looks like. So if you're ectomycorrhizae, you're between the cells. If you're arpuscular mycorrhizae, another word for arpuscular is Hastoria. Here's an arpuscular or a Hastorium. Right, you're branching out. You can see in this picture here, these are all plant roots or plant cells, and all these are the fungal or in this case, not called Hastoria, but the arpusculars. See, and these things penetrate the cell wall, but they don't go through the cell membrane. That's the important thing, folks. If you're penetrating the cell membrane, you're probably parasitizing. These guys aren't parasitizing; they live in a mutualistic relationship. But they can penetrate into the cell wall because what they do is they actually get small, very small. See, the ectomycorrhizae stay a little bit bigger, but the arpuscular mycorrhizae, they are bigger, then they get smaller and smaller and smaller until they can finally kind of squeeze in between the cell membrane and the cell wall. Gives them more access to the nutrients again. Okay, very cool, right? Now, you want to see something really going to blow your mind. We just went through this whole thing about alternation of generation and a gametophyte and a multicellular sporophyte and which is what. And you guys did a whole lab on which generation gives rise to what. You know, at the time we talked about how much different that is than animals, right? We only make a single-celled gamete thing. We make a zygote. We don't make a multicellular one. And now I'm going to throw another, I don't know, sort of not detour, but difference that occurs in reproduction, right? Reproduction is a characteristic of life. However you do it, asexually, sexually, a lot of offspring, few offspring, no care, little care, however you do it, is kind of up to whatever keeps you alive, whatever gives you fitness. So this is what's been working for fungi, for most of the fungi. So let's take a look and see how this works. I'm not even sure where to start. Let's start with the zygote. Let's go over here. First of all, the zygote is the only diploid stage. You see, as I said, in fungi, it isn't really about the cell, even if you are septate. That's why they have those pores. It's really more about the nuclei. Those nuclei can very easily pass through those pores of that septate hyphae, those little openings there. So the only time a diploid form is produced during fungal reproduction is when we have this very short-lived zygote. And there's a little bit of terminology here. But when a zygote is formed, whether we're talking about a fungus or really anything, when you were formed as a zygote, when sperm and egg mix together, what basically is going on is a process in which first the cells are going to fuse. This is called plasmogamy. Just put a mark by it. I was trying to make a 1, but that looks really bad. And then skip what the middle part is for just a second, the heterokaryotic thing. But again, and then after the sperm and egg have fused, then the nuclei fuse, and that's karyogamy. That's a better number 2. That happens in all cells, right? Plasmogamy, karyogamy. In our case, plasmogamy to karyogamy happens pretty much instantly. As soon as the sperm penetrates the egg surface, it deposits its nuclei. As soon as it becomes fused with the egg, the nuclei fuse. So it's very quick. In other organisms, that ain't necessarily the case. Plasmogamy may actually occur, and you don't get karyogamy, see in the red, for a while. Hours and days, centuries. Okay? I'm going to go ahead and continue on here. I know I said I was going to start with the zygote, and I kind of moved backwards, but I think this is a good chance to tie all this together because of where we are. So look at the picture of the color of the arrow. It's purple, right? It's not the red of diploid or the blue of haploid or whatever color you want to call it. It's more of a purple color. It's called a heterokaryotic stage. Now, a heterokaryotic stage means that if you're a septate hyphae, in those little sections, you actually might have two haploid nuclei. They might be fused, but they may not be. Each cell would be in a heterokaryotic stage. Specifically, if you're septate, we're going to call you dikaryotic because you have two haploid nuclei in a cell. Okay, now, that whole heterokaryotic definition, having many nuclei if you're synocytic, the ones without the septate, the hyphae that have no break between the cells, then you're all kind of heterokaryotic. Again, it's not about the cells. You're just full of heterokaryotic. And when we get to reproduction of specific types of fungi, I'll emphasize again what that's doing, how that's important. But overall, being heterokaryotic does have an advantage. It's kind of like the advantage of being diploid. It's not the same as diploid, and I mean that in a way that I don't mean that in a better or worse way. There's a 1.5 million species, folks. There's a lot of fungi out there, so whatever this heterokaryotic stage does for these organisms works very well. Remember, there's even though being haploid gametophyte means you got to be small and kind of delicate, there's a lot of moss out there. The diversity, even the biomass of moss, there's quite a bit. So you got to know that this is not a better or worse thing. But if you're heterokaryotic, you get some of the advantages of diploidy. Now, we talked about being the advantages of diploidy back during the genetics chapter or during the evolution chapters. Does anybody remember what the advantage of being diploid could possibly convey? There you go. Thank you, Spencer. Right. You got a backup piece of DNA. Now, I understand all of the dominant and the recessive that we talked about plays into all of that. You can get two recessives, and many times that's not a good thing. But with all the different probabilities of things that's happening, having another set of DNA, as Spencer put, right, a backup piece of DNA is not a bad idea. Now, heterokaryotic organisms have come up with the same idea. If you get a visual of it in septate organisms that are dikaryotic, right, one cell, two haploid nuclei, they're the same as being diploid. What's the only difference? My pen doesn't work. What's the only difference? They haven't fused the nuclei. They haven't done karyogamy. But they still have two sets of genetic information. So it's kind of like being diploid. And I don't know enough about the heterokaryotic stage, but I wonder if the old dominant and recessive thing plays out when you're heterokaryotic. You know, if you're diploid like we are, if you have the recessive allele, it doesn't get read. That's why it doesn't express, unless that's all you got. But I wonder in heterokaryotic. It's a good thing to remind us to look up. Heterokaryotic. I'm going to write the word here. Do we still get the dominance and recessive? Good question. Good question, Steve. 2 points on the next exam. All right, now, so everybody understands heterokaryotic, what it is, why it happens. Let's jump to the diploid stage, the zygote, which is short-lived. What's the purpose of the zygote? And what I mean by purpose, take a look at what the zygote, I mean more like what the purpose of what the zygote does. What's the purpose of what the zygote's doing there in meiosis? What are you getting out of meiosis? Drum roll. Meiosis gives you... There you go. Thank you, Thomas. Gives you variability. All right, so you got spores. Now, these are sexual spores, and that's going to, I put that word in there because there are also what we call asexual spores. If you keep following around a spore when it germinates and then eventually becomes hyphae, like a string of hyphae and then a bunch of hyphae and a mycelium, there's also asexual reproduction, and they produce spores too. It's much easier, right? Mycelium, spore-producing structure. Oh, you know what that is? Toadstool. Toadstool is a spore-producing structure. Look at that toadstool. It's haploid. So people say, oh, critters are diploid, and you point to a fungus, that's haploid. You point to a moss, you say that's haploid. Haploid ain't necessarily the only game in town. But the spores you get out of, if you're doing asexual, those are asexual spores. Okay? And asexual spores can't be produced by meiosis. Let that sink in for a second. Can't divide N in half. So the way you're going to get asexual spores is you're going to do it by mitosis. So whereas down here you get variability, what do you get up here when you reproduce your spores just by mitosis, when you do it asexually? Drum roll. We get Nicole. What do we get? Sydney, what do we get? Marcelo, Jennifer, what do we get when we do mitosis with these spores? When we undergo asexual reproduction, we get variability here. Asexual spores, what do you get? You get lots. Lots and lots and lots and lots and lots and lots and lots. You get numbers, you get numbers, you get numbers. And of course you're getting numbers because you know most of them aren't going to make it. All right, so back to the loop here in the middle. These spores, whether they're sexual or asexual, begin a mycelial mat. Now, one day they want to have kids of their own. So what we say is they attract each other. These mycelial mats, the actual hyphae, they're not sexes. We're going to call them mating types, folks. But they attract each other. But they kind of use, at least in some people's view among humans, but in animals for sure, and among insects, they use this thing called pheromones. Pheromones are like external hormones. Here we are back external again, right, with the exozymes. But fungi or organisms, not just fungus, can secrete these chemicals into the environment, and it signals a measure of sexual receptivity. So among sexually reproducing organisms, a female would secrete this chemical to let the males know around that she's, in a reproductive cycle. Not all organisms reproductive cycle the whole time we do, or all year round. Wolves, for example, with twice a year or once a year. I think it's once a year wolves do. So do you know, Thomas? Is it once or twice a year? I think dogs are twice a year, wolves are once a year. I think that's one of the differences between them. I don't remember. But anyway, whenever you do it, the female's got to know, or the male's got to know. Right? Most of the time, she won't have anything to do with him because he's probably going to get in the way and try to eat one of her kids, so she keeps him away. But you secrete these pheromones, and it's like an attraction thing. Yeah, mine too. It's always the best thing. It prevents testicular cancer if you're not going to breed the animal, that's for sure. Unless it's female. It also prevents cancers in females too. But these pheromones of these different mating types, of these different strands, attract one another. That also prevents them from inbreeding. So it can do a disattraction. And when those two little things come together, guess what? They fuse their cells, plasmogamy. And then in a little while, or a little longer, or maybe a long time from that, they fuse into karyogamy. All right, so during the time when you're in this heterokaryotic stage, you might think that you might be tougher, stronger, okay? And because you have an extra set of genetic information, you got a backup set of genetic information, like Spencer said. So if you think about that and do a little bit of detective work, you might be able to, understand when, as we go through the different reproductive modes, you might be able to understand why these organisms do it, when they become heterokaryotic. All right, we'll get to that in just a second. Let's go to the easy one first. Let's go to asexual reproduction. There are many organisms that are sexual and asexual that can do both. Some prefer one over the other. Some prefer to reproduce sexually versus asexually, but many of them can do it both ways. There are a group of species out there that have this artificial sometimes I hear it as a taxonomic name. So again, go back to this taxonomic idea that, you know, the names are a little bit loose. But sometimes I've seen phylum, right? That's the plural of phylum, right? In plants and in fungus, sometimes I've seen phylum referred to as a division. So it doesn't go kingdom phylum, it goes kingdom division. So if you see that word, it's not used consistently. But speaking of not using consistently, the word Deuteromycetes, the one in the black, the bold, that is an artificial phylum or an artificial division in a lot of people's classification schemes. It's artificial because we classify fungi based on the way they reproduce. Right? You'll see what I mean in just a minute. But if we don't know the way an organism reproduces, for example, we haven't witnessed it yet. A lot of these organisms stay asexually reproducing, and it could be like for long periods of time. We're not sure if maybe they never do or maybe they just haven't yet. But we throw them into this collection called the Deuteromycetes, which is translated sometimes as the imperfect fungi. It doesn't mean there's anything wrong with them. It means we don't know how they sexually reproduce. And if we find out how they do, then we stick them into the right group after that. Okay? Now, members of the fungal group that not necessarily Deuteromycetes, but then undergo asexual reproduction are molds. Molds undergo asexual reproduction. That's the black red mold, the gray mold that gets all over your orange, right? In some cases, it's actually, in this case, this is penicillium. See the penicillium, oh, so cool. When we look at it under a microscope, it looks like the Greek mythological, it was a female, right? Had snakes out of her head. Medusa. It looks like a Medusa. And each of these little Medusa, each of those blue-green things is rings of spores. See, these are the asexually produced spores. Lots and lots and lots. If you're yeast, while I'm drawing this, you can see on the right the way yeast do, they just bud. Hey, bud. They just split. They just asexually reproduce right there, see? And they can do that pretty quickly, too. They just reproduce by fission. This, by the way, is Saccharomyces cerevisiae. What do you think this yeast is used for? There's more than one species of yeast, but this yeast is a popular yeast used in what process, do you think, based on the name? Baking bread. You do use yeast for baking bread, but I was thinking this particular species right here, based on its name, you know, what do you think is often done with that? If fungus are so into mucilin, why don't? If fungus are so into mutualism, why do they create antibiotics? Not all fungi are into mutualism, Spencer. Right? Not all. I mean, the mycorrhizae are, the lichen, the fungus that's in a lichen, it is. But, you know, like the predation on that nematode, that definitely wasn't mutualism. Right? So I'm trying to think if fungus are so into, does that make sense? Now, the antibiotic part, organisms, including fungi and ourselves, are feasted upon by bacteria. Bacteria love to take advantage, especially when your immune system is compromised. So everybody needs antibodies. Every living thing really needs some way to defend themselves against those pesky little bacteria. Thomas, you were saying Saccharomyces cerevisiae is the kind of yeast we use in baking bread. No, it's a similar process. I don't know if they use this. They might use this yeast in baking bread, but they also use it in another process that the name seems to be more indicative of. None of you have taken Spanish class? None of you have ever been to Mexico or into any Spanish-speaking? Okay. No? So if you were sitting at a bar in one of the Spanish-speaking nations, like in Cuba, my wife is Cuban, and she wants to go back to Cuba someday. I'd love to go back. I think it'd be great. They got great diving. And you said to the bartender, "Uno más cerveza, por favor." What would you be saying to him? Yeah, beer. So what is this yeast used for? Isn't that amazing? Isn't that amazing, Jennifer? How you didn't click until I said the sentence. So it's like you said the sentence before, but only knew that a beer came your way, not that you were specifically asking for a cerveza, right? I get it. I remember overseas when I was in the Philippines or in some of the other countries we went to, we learned very quickly how to say beer in whatever language was there. Okay, whatever language that was the first word we learned. A beer and then restroom. That's the first word we learned. Yeah, Saccharomyces cerevisiae is the one we used for the brewing of beer. Okay, I think biologists sometimes like to have a little bit of fun when they name things. Where am I? I'm right here. Okay, ancestry. Where did they come from? Well, like I said, they're actually more closely related to you and I. They don't fossilize very well for reasons that I've gone over quite a bit. They don't fossilize very well, but we do look in that molecular clock. You remember that molecular clock says genetic change occurs on a regular basis. Tick tock. Like what's that thing that music kids use that goes back and forth? Tick tock. I love the way they sound. I'd like to get one from the house. Tick tock. What is that called? Didn't you guys ever take music? My friend played. Yeah, that's it. Right. So that's the molecular clock. Tick tock. On a regular basis, it helps you keep in time when you're playing music. But by that analysis, we know that animal and fungi had an ancestor about 1 billion and a half years ago. So it wasn't long after the plants, but it was after the split between. So it like. Are we showing it here? Yeah, I don't want to confuse you. So first of all, the ancestor was aquatic, unicellular, flagellated, and guess what? Was a protozoan. All right, so the animals and the rest of their protozoan relatives went off this way, okay? And what's not included in here is one of those protozoan relatives that they're not showing is, of course, the plants. That's also a protozoan relative. You're related to plants through the fact that we were all protozoans at one time, whatever that means. And the nuclearids right here, that's actually not a fungus. That's a protozoan too. Okay, so you see once again that the animals and the fungus, we had a common ancestor right about here. And sometime later on, a protozoan relative, specifically the plants, split off. So we're not by much, but technically we are more closely related to a fungus. And it's the chitin thing that's very interesting. Okay, fungals and animals are, and I put closely in quote, or in italics, of course. We form a group of organisms in which we have a rear end flagella. Your flagella can be on the front, like if you're a euglena, or it can be like behind, like the sperm. Isn't that cool picture that looks like a computer generated? But we have posterior flagella. If you have a flagella, okay, in some of your cells, and this is going to obviously be more likely in males, right? Your sperm having the flagella in the back. So that's us and the animals, but the fungi is actually more closely related to the nuclearids than it is to the animals. So even though fungi and animals are more closely related, the fungus is actually more closely related to another protozoan. And on the other side of the group, in the animals, there are also protozoan relatives. See how screwed up the protozoan group is? So you don't even think about the kingdom protozoan anymore. Protozoans are mixed in everywhere. Like I said, they were the stem group. They kind of gave rise to everything. All of us owe our existence to the protozoans. But all right, here's the diversity part. So up here somewhere would be the animals, and a little further down would be the plants. Okay, just to reiterate that one more time, and then I'll move on. Small little group of species called the chytrids. The chytrids, there's a chytrid. See, little hyphae. The hyphae is what makes them chytrids. And I'm going to go back and forth to the screen, so don't worry if you haven't gotten everything. So the chytrids are about 1,000 species. The phylum Chytridiomycota, sometimes called division Chytridiomycota, found all over the place, even down by those hydrothermal vents. even though they're down where that place where we think maybe biogenesis occurred. They do all kinds of things. They act as decomposers and parasites, mutualists. Mutualist and the parasite, the only difference between the two is that a parasite, like this one up above, this chytrid, is parasitizing an Odogonium. Odogonium is a green algae. Okay, so the chytrid is actually growing in with its hyphae into the cell membrane and parasitizing that. So parasitism, somebody's got to lose, and mutualism, right? Once again, both of you benefit. These guys came out very early because even when we look at the spores now today, we find that they still have zoospores. Now, zoospores, that's a little bit misleading. The zoo means animals. You go to a zoo to see animals, okay? Zoology is the study of animals, and that's the way it's pronounced, by the way. It's not zoology. And I always forget, here I am giving you a lecture lesson, and I forget. I think it's the second O that's umlauted. But that makes the word zoology, not zoology, or maybe it's the first O. I forgot my German. But anytime you see like the word zoospore, it means it's a mobile thing. So we associate the word zoo with what animals are usually known for. They can move, okay? So this little spore down here has a flagella. Notice at the rear end, so it's a posterior flagellated zoospore. That's what a spore of a chytrid looks like. Now the next group you guys are really familiar with. Not a very big group. It's about 1,000 species, but this is the zygomycetes, okay? And the zygomycetes are the like the black bread mold. The mold that grows on strawberries only seems like a day or two after they come home from the store, right? Like the ones you see in the upper right. This is, like I said, it's a diverse, but it's not a very big group. Diverse in terms of number of species, well, it's a very different group, but it doesn't have a lot of species. In other words, the diversity that's there is very diverse. Okay, this is what we call things commonly as the molds. They also exist as parasites and commensals. That's another symbiotic relationship. Remember, commensal is a plus and a zero, like the egrets and the cows. Where are we in time? We have 5 more minutes, right? We're doing fine. We won't finish this chapter, but we'll be all right. They have synocytic hyphae, so they can grow very quickly. Synocytic hyphae don't have the septate. Okay, I've already illustrated, or I've already introduced you guys to the overall scheme. Notice that we have a sexual reproductive mode, and that's when the environment's deteriorating. Okay, remember I said that they sometimes do it when it's great, sometimes when it's deteriorating, sometimes they vary. However, in this group of black bread mold, they typically undergo asexual reproduction. So they produce spores that simply germinate and produce mycelia, that produce spores. These things called sporangium. You'll see them in the microscope. They look like lollipops. This is rhizopus growing on bread. See the lollipop? Some of them actually have sporangium they can direct. I don't remember the name of this one, but this one is facing its sporangium in a way, and it's going to eject its spores in a way, in a particular direction. Isn't that freaky? Looks like from a sci-fi flick. Okay, so I'm not going to spend a whole lot of time going on what's going on through here. I just want to stop during the heterokaryotic stage. During the heterokaryotic stage, something called a zygosporangium forms, and you'll see it under a microscope, but you can even see it here. It's very harsh looking. It's lumpy, okay? It's metabolically inactive. It's like a seed. It's not dead, but it's metabolically inactive, but it's very resistant to freezing, drying. It's very resistant to a deteriorating environment. Okay, now that's going to stick around, and when the environment begins to appear like it's going to become better or starts to become better, then karyogamy is going to occur. All right, so you get this short-lived nucleus only to turn around and produce these variable spores. So you get this zygosporangium waiting things out, and as soon as the groundhog, I always forget whether he sees the shadow or not, as soon as the sun comes through the clouds, it says, all right, let's crank out a zygote. Let's put these nuclei together. Boom. There might be many of them that are, because you're synacitic hyphae, right? You got all these nuclei. Boom. They start combining together. You get all that variation, and then you're going to go through meiosis. You get a ton of that variation as well. World's getting good. We should find out if there's a place where we can live. All right, let me do this last group, or not last group. Let me do this next group, and then we'll finish. All right, this is the group called the Glomeromycetes. This is a small group, only about 160 species. I forgot to go to that other screen. There we go. About 160 species. About 160 species, but some of those species are very important species. So they were once put in with the black bread molds, but nearly all of these, this is the important part, nearly all of these form what we call arpuscular mycorrhizae. Remember I said there was arpuscular and there was ectomycorrhizae on the outside. Most mycorrhizae, and the best type of mycorrhizae, is arpuscular. Over 80% of the plant species, over 90% of plant species have mycorrhizae, and then of them, over 80% have arpuscular. So again, to remind you, there's the plant root, there's the hyphae, and this is a really cool picture. This is the arpuscular. Okay, it looks like the little haustorium. It looks like something kind of exploded and burst out, right? And of course, what that is increases surface area to volume ratio again. And and notice what it's inside of. It's not inside of the cell. I don't know where the cell is exactly in the picture, but it's inside of the cell wall. It's not parasitizing it. Okay, folks, I'm going to stop for there. Let me stop the recording.